Hello, my name is Dr. Robert Daum. I'm a professor of pediatrics and microbiology at the University of Chicago Medical Center. I'm the principal investigator and the director of the MRSA research program that we've established at the University of Chicago. When we first realized that MRSA infections were occurring in children in Chicago that required hospitalization at an alarming rate that had increased dramatically in a few years, we began to feel that we needed to publish the findings. And when we tried to do so, there was resistance to the idea that this new event that we thought we were observing was in fact correct. The resistance occurred because the isolates that we saw in the community were susceptible to an antibiotic called clindamycin, which MRSA isolates usually weren't. And the thinking was that we probably didn't understand that these infections truly were MRSA, and we were probably wrong in that regard. But we demonstrated that the gene that was the signature gene for MRSA, MEK-A, was in fact in the strains in the community, and so that part of the resistance was at least the first step was overcome by recognizing those two facts. The signature gene is MEK-A, and with the two exceptions in history, all MRSA strains have the MEK-A gene. But the fascinating thing that is still not particularly understood is that the MEK-A gene comes to the staph organism with flanking DNA, integrates into the chromosome in precisely the same place, and the events associated with the integration and the excision of this DNA cassette, which is now called the staphylococcal chromosomal cassette MEK, or SCC-MEK, these events are not completely understood, and they're very interesting. The Japanese group, led by Dr. Hiramatsu and Dr. Ito and their colleagues, had defined SCC-MEK elements that were big and kind of clunky and not easily mobilized. The bigger the chromosomal insert, the less mobile that it is. And so we collaborated with them to share some of our community associated strains with them. And together we discovered a new SCC-MEK variant called SCC-MEK type 4. They had found 1 through 3 in healthcare associated strains. And SCC-MEK4 distinguished itself by having the MEK-A gene, of course, by inserting into exactly the same place in the staphylococcal genome, by presumably being able to integrate and excise by a poorly understood method, and transfer from strain to strain by a poorly understood method. And it's this transfer that gave you the sense that this cassette was promiscuous, and that is able to transfer itself to methicillin-sensitive strains, convert them into MRSA strains, and thus we had a new clone in the community that was in fact a MRSA clone. So this SCC-MEK type 4, and there's now type 5 as well, with a very similar kind of story to it, are very important things to understand in terms of how this community MRSA event epidemic arose at the molecular level. The collaboration that we've undertaken with the group in Japan has been followed by an effort in our laboratory. We've been very active and among the leaders, I think, in terms of understanding the molecular features of the SCC-MEK element type 4, characterizing a new variant of the type 5 SCC-MEK element that appears to be unique in terms of its circulating and causing community-associated MRSA infections in Taiwan. We were the first group that sequenced the SCC-MEK5T element that circulates in Taiwan. We also have a very elaborate team in our lab led by Dr. Susan Boyle-Vavra, where we're able to characterize with PCR techniques and sequencing techniques variants in the type of SCC-MEK element present in the strains and do molecular typing of these strains. So that we've also been able to understand, start to understand how USA 300 has become so important by looking at molecular expression of Staphylococcus aureus virulence determinants and have made a lot of progress in this regard by understanding that the paradigm of acquisition of one new virulence gene by USA 300 is probably not the explanation of how it became so prominent, but rather it's been looking at the expression of virulence factors that has explained USA 300's prominence. This is a embryonic work that Dr. Boyle-Vavra, Dr. Chris Montgomery, and other members of our lab team have been really spearheading.
My name is Dr. Michael David. I'm joining the faculty at the University of Chicago in order to continue my studies on the epidemiology of methicillin-resistant Staphylococcus aureus. The most interesting thing about this diversity of strains in different parts of the world is that the strains that typically cause MRSA infections in healthy people in each continent and in each country tend to share certain characteristics in their genes that are not shared with strains that cause infections in hospitalized patients. So it's a really important and fascinating but subtle distinction to be made between community-associated MRSA strains and healthcare-associated MRSA strains. That community-associated MRSA strains, even though the strains differ in different continents and in different countries, they all tend to carry one certain type of toxin gene called the PVL toxin gene, and they all tend to carry the same kind of cassette in their genes that called SCCMEC4 that makes a strain of Staph aureus, a bacterial strain, into MRSA. Why these different strains share these same few genetic characteristics but don't share the same strain type is something that's going to require a lot more research to understand. Several researchers in Europe, particularly in the Netherlands, and more recently in the United States, in Iowa and in Canada, have found that livestock, especially pigs, not infrequently carry MRSA strains in their noses. These MRSA strains are most commonly a specific strain of MRSA that is known as ST398. In their studies in Europe and in the United States, researchers have found that farm workers who are commonly in contact with these pigs in the course of their work also can become colonized or can become asymptomatic carriers of ST398. There have been a number of case reports of infections with ST398. Most of those case reports have been in Europe, but very few of those infections have been severe or invasive infections. It's not clear yet how important ST398 will be in the epidemiology of MRSA in the community, so it's unclear whether they will become important for the general population. So far, they haven't shown themselves to be very common pathogens, but it's an area that requires much more research.